Okay, let's do the whiteboard version of smell. There we go. GPCRs and smell. All right, let's try this. So, first of all, this is going to be taking place at a spot called the olfactory neuroepithelium. Olfactory neuroepithelium. Yeah, there we go. Which is just a little tissue right up in the top of your nose. It's the thing they end up swabbing when you have the flu test that goes up your nose or the COVID test that goes up your nose. So, yeah. All right, and so on this part of the body, you've got some cells that are right there on the surface, and these cells have these wonderful molecules in them called odorant receptors. And odorant receptors go through the membrane 7 times. So 1, 2, and then they have an extra little helix here. And each of these parts is an alpha helix. So I'll draw a little squiggle over them so you can tell. Pretty much always when a protein crosses the membrane, almost always, it's going to cross in an alpha helix. There are a couple where it makes like a giant beta-stranded barrel that goes through the membrane, but I know those from bacteria. I'm not even sure there are any in the human proteome, which is all the proteins a human can make. All right, so there we go. We got our odorant receptor, and this particular odorant receptor is going to bind a molecule, and so this is going to be one of those molecules from cilantro. And so we'll put a little 1, 2, 3, 4, and an oxygen. Let's see, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. So that would be decanal or undecanal, or there are all those other versions of it, something like that. That's probably a little hard to read, but you can pull it off the actual slide set. And so we got a little molecule, and that is going to go in. And so let's draw a little arrow that shows that this is going into our odorant receptor. And so once it goes in, then the odorant receptor is going to change. And so when it changes, it's going to be pinched a little more like this. 1, 2, 3, 4, and then 5, 6, 7, and then 8. So there we go. There we go. That one's going down behind. All right, and so we're going to change the position of alpha helix 5, 6, because it's attached to 5, and 8. These are going to move, and 8's going to go upward, and 5 and 6 are going to sort of tilt outward. So there's 5, there's 6, and there's number 8. All right. Great. And so now we've got the potential to activate something. All right. So now that we've changed the position of these, we can interact with the G protein. And so our G protein coupled receptor, which is our odorant receptor, which is a GPCR, G protein coupled receptor, is going to interact with a G protein. So we'll draw our little G protein here. So it's got 2 subunits that are going to have, actually let's make it a different color. Let's make it a nice blue color. There we go. All right, so we're going to have an alpha subunit, and we're going to have a beta subunit, and we're going to have a gamma subunit. About like so, and they're going to be all stuck together. All right, so show those actually kind of merge together. There we go, about like that. There, that's better. That's better. All right. And the alpha subunit, when it comes in, is going to be holding on to, of course, GDP, because our G protein needs some help. So this is a G protein with GDP. So it is the inactive version. And the next thing that's going to happen is this is going to act like a little can opener. You can think of it as that, and it's going to pry that GDP out and allow a GTP to come in. So the next step is that, here we go, we're going to say GTP in and GDP is out. And so we're going to end up with a, let's just change it on here, but I suppose we'd redraw it if we were doing this for a test answer. So now let's put in a GTP. And so now it's turned into the G protein with GTP, which is going to be active. Oh, that's some terrible handwriting. All right. All right, got an active G protein, great. And so now, at the same time as it is picking up the GTP and activating, it is going to break apart into two halves. And so it's going to split right here. Okay, and the beta and gamma are going to go down here. So G protein, beta and gamma go to the nucleus and inhibit expression of other odorant receptors. Yeah, which is basically what's going on there. All right. So great, we've done that. So you can draw the little genes, and so here's odorant receptor 2, odorant receptor 3, and up here can be odorant receptor 1 because we made it black up here. There we go, odorant receptor 1. Make sure I'm still on screen. Yes, I am. All right. And so beta and gamma are going to land here, and they're going to put a stop to that. So this is our way of showing that we are not going to transcribe that, and we are going to definitely transcribe more of odorant receptor 1, which is going to go up here, and boom, make more of that. And so this is the process of sensitization. Or we're just going to make more odorant receptor 1 after we get a hit on it. Yeah. All right, fantastic. So now, we've got our G protein, and it's split apart. And so let's make a new little thing. Let's make it over here. So let's make some more membrane up here, because the alpha subunit is still stuck to the membrane. There we go. Okay, and so here is our G alpha olfactory, and yes, we should put OLF by these guys, OLF, OLF, because it is a olfactory version. So it's actually a GOLF, G protein. Okay, G alpha olfactory, and this one is still going to be holding on to GTP, so it is the active version. And so that's coming from over here. There we go. All right. Now this one is going to interact with this enzyme, and we need another color for this enzyme. Let's see if we can make it orange. Let's see if that shows up. It doesn't show up super well. We'll make it orange outside and black inside. I don't know, we'll go pumpkin colors. Why not? Yeah. Okay, and so this is adenylyl cyclase number 3. Adenylyl cyclase 3. Yeah, that's the edge of the world. AC3. All right. And so what this adenylyl cyclase 3 is going to do is it's going to take in ATP and it's going to spit out cyclic AMP. All right, and now we'll send our cyclic AMP back over, and let's do it over on this side. We got space? Yeah, we got space. All right. So meanwhile, there were a couple of ion channels. Let's draw our ion channels. So here we go. There's channel number 1, and here's going to be channel number 2. Okay, and so channel number 1 is, of course, the cyclic AMP gated calcium and sodium cation channel. Ion channel. There we go. I mean, they're ions. I don't know. That's kind of implicit. And so Ca2 plus and Na plus are going to go in here. Boom. And they're going to come through. So we've got Ca2 plus and Na plus coming inside. And this is gated with cyclic AMP. Boom. So cyclic AMP is going to hold the channel open. Yeah, all right. And so this increases the positive charge inside the cell. Right, so we're trying to charge up this cell basically. We're going to make it super positive. All right, and then on the other side, we've got another channel that is calcium gated. So let's draw a little blue calcium here. Boop, boop. So Ca2 plus is right there. Holds the channel open. And so this is a calcium gated chloride ion export ion channel. Yeah, an ion is kind of redundant there, but what the heck. Okay, and so we've got some Cl minus on the inside. We'll draw a green like chlorine gas. Cl minus. Do we see that? Barely. And so the Cl minus is going to go up, up, and away. Cl minus is now going to move outside. And so we are again going to increase positive charge inside the cell. C-E-L-L. Okay. Yeah. And then somewhere, you're just going to tell me that once the cell is positively charged enough, voltage gated ion channels will carry the signal via neurons to the brain. Something like that. There we go. And put all that together, that's a fairly complete answer. And yeah, we got a nice process there. We got all the parts of it. So I don't know. See what you think. Yeah, yeah, a lot of stuff. But that'll be a real good test answer. And yeah, these sorts of channels and cyclic AMP and stuff like that. This is stuff that's going to come back hard in cell biology. You're going to be learning little networks like this, because that's how the cell works. It's like one thing activates another thing and another thing. Nothing just directly does an entire job itself. It's always these big, long chains. So, all right. Thanks very much. Hope you enjoyed this one. Hope it's useful. Yeah, I am out.